الو الو. تيست. الو. ضبط ضبط. السلام عليكم. لو سمحتوا الجميع في أماكنكم، لو سمحتوا بنبدأ بعد شوي. شكرا. بسم الله الرحمن الرحيم. معالي الأستاذة الدكتورة رحمة بنت إبراهيم المحروقية الموقرة، وزيرة التعليم العالي والبحث العلمي والابتكار، أصحاب المعالي والسعادة، الأفاضل أعضاء مجلسي الأمناء والإدارة، أعضاء الهيئتين الأكاديمية والإدارية بالجامعة، الطلبة الأعزاء، الضيوف الكرام، صباح الخير وأهلا وسهلا بكم جميعا. بالأصالة عن نفسي ونيابة عن جامعة ظفار ووزارة الثروة الزراعية والسمكية وموارد المياه، يشرفني أن أرحب بكم في الملتقى الدولي الثالث حول اللبان والنباتات الطبية، المنعقد تحت الرعاية الكريمة لمعالي الأستاذة الدكتورة رحمة بنت إبراهيم المحروقية، وزيرة التعليم العالي والبحث العلمي والابتكار.
بسم الله الرحمن الرحيم. Your Excellency, Professor Rahma bint Ibrahim al-Mahrouqiya, Minister of Higher Education, Research and Innovation, Your Excellencies and Highnesses, Honorable Board of Trustees, Honorable Board of Directors, distinguished guests, respected colleagues, our dear students, a very good morning and warm welcome to all of you. On behalf of Dhofar University and the Ministry of Agriculture, Fisheries and Water Resources, it's my great honor to welcome you all to the third international meeting on frankincense and medicinal plants, held under the esteemed patronage of Her Excellency Professor Dr. Rahma bint Ibrahim al-Mahrouqiya, Minister of Higher Education, Research and Innovation.
ويسرنا في هذا المقام أن نتوجه بخالص الشكر والتقدير إلى الرعاة الكرام الذين أسهم دعمهم السخي في إنجاح هذا المؤتمر، وهم الرعاة الرئيسيون، جامعة ظفار ووزارة الثروة الزراعية والسمكية وموارد المياه، الرعاة البلاتينيون، غرفة تجارة وصناعة عمان فرع محافظة ظفار وبيت البخور العالمي الصين، الرعاة الفضيون، لبان ظفار وشركة مكة للتجارة. نعرب لهم جميعا عن بالغ امتناننا وتقديرنا لالتزامهم ومساهمتهم.
We wish to extend our sincere appreciation to our generous sponsors whose valued support has made this conference possible. Our main sponsors, Dhofar University, Ministry of Agriculture, Fisheries and Water Resources. Our platinum sponsors, Oman Chamber of Commerce and Industry, Dhofar Branch, Global Incense House, China. Our silver sponsors, Luban Dhofar and Mecca Trading. We are deeply grateful for their commitment and contribution.
أيها الحضور الكرام، استعداداً لانطلاقة هذا الحدث المهم وتسليطاً للضوء على رؤيته وأهدافه، ندعوكم الآن لمشاهدة فيلم وثائقي قصير.
To set the stage for this important gathering and to highlight its vision, we invite you now to watch a short documentary video.
تمثل شجرة اللبان شعاراً لمحافظة ظفار نظراً لأهميتها التاريخية والاقتصادية، وتعد من أهم المعالم السياحية والتاريخية للمحافظة، حيث يحرص معظم الزوار والسياح على شراء اللبان كالبخور الذي يباع في مختلف الأسواق التقليدية والتجارية. تتواجد أشجار اللبان في المنطقة الواقعة خلف الجبال التي يطلق عليها ظل المطر، إلى جانب المنحدرات المنخفضة وفي قاع الأخاديد والجداول، وبكميات أكثر كثافة في بطون الأودية العريضة الأكثر حجماً. محمية جبل سمحان الطبيعية وسهولها ومنحدراتها تحتوي على أكثر من نوع مثل اللبان الحوجري والحاسكي، والنوع آخر يسمى الرسمي، وينمو في سهول ولاية سدح، المنحدرة من جبل سمحان. يبدأ إنتاج وجني محصول اللبان في محافظة ظفار خلال منتصف مارس وحتى يونيو من كل عام باستخدام الطرق التقليدية المتبعة والمتعارف عليها منذ القدم، بينما تبدأ المرحلة الأخرى بعد فصل الخريف مباشرة حتى دخول فصل الشتاء. محصول اللبان في محافظة ظفار يشتمل على عدة أصناف مختلفة من ناحية الجودة حسب التوزيع الجغرافي، وأجودها الحوجري والحاسكي والنجدي والشزري. وبهدف إيجاد هوية عمانية ذات بعد عالمي، قامت العديد من الشركات بتصنيع منتجات متنوعة من اللبان العماني كالزيوت الطبيعية وماء اللبان المركز التي تستخدم في مكونات الأدوية والعطور والصناعات التجميلية العمانية. ويمتص النحل رحيق أزهار شجرة اللبان لينتج عسل اللبان العماني النادر عالمياً. جهود حكومية كبيرة تبذل للحفاظ على أشجار اللبان العماني لأهميتها التاريخية والمستقبلية. حيث قامت وزارة الثروة الزراعية والسمكية وموارد المياه بجمع بذور اللبان من مختلف المناطق الجغرافية المعروفة، إلى جانب إنتاج شتلات من هذه الأصناف بمشتل الموارد الرعوية بالمديرية العامة للثروة الزراعية والسمكية وموارد المياه بمحافظة ظفار. محمية وادي دوكا التابعة لوزارة التراث والسياحة، والتي أدرجت ضمن قائمة التراث العالمي الثقافي والطبيعي، أنشئت للمحافظة على أشجار اللبان، وتحتوي المحمية التي تعتبر البيئة الملائمة لنمو أشجار اللبان على أعداد كبيرة من أشجار اللبان إلى جانب الأشجار الطبيعية المعمرة. وتمتد كثافة أشجار اللبان على مساحة تقدر ب5 كيلومترات مربعة. تحتوي المحمية على أكثر من 5000 شجرة لبان مزروعة وطبيعية.
أيها الجمع الكريم، يسعدنا الآن أن ندعو الأستاذ الدكتور عامر بن علي الرواس رئيس جامعة ظفار لإلقاء كلمة الترحيب، فليتفضل مشكورا.
Now it's my distinct pleasure to invite Professor Dr. Amir bin Ali Al-Rawas, the Vice Chancellor of Dhofar University, to deliver the welcome speech.
بسم الله الرحمن الرحيم. الحمد لله الذي أودع في الطبيعة أسرار الشفاء، وجعل في خيرات الأرض ما يغذي الأبدان ويحصن الأرواح، والصلاة والسلام على سيدنا محمد المبعوث رحمة للعالمين وعلى آله وصحبه أجمعين. معالي الأستاذ الدكتورة رحمة بنت إبراهيم المحروقية الموقرة، وزيرة التعليم العالي والبحث العلمي والابتكار راعية الملتقى، أصحاب المعالي، أصحاب السعادة، الباحثون والخبراء والمشاركون الكرام، أيها الأعزاء، السلام عليكم ورحمة الله وبركاته. يسعدني ويشرفني أن أرحب بكم جميعا في رحاب جامعة ظفار، ونحن نفتتح معا أعمال الملتقى الدولي الثالث حول اللبان والنباتات الطبية في محافظة ظفار، الذي تنظمه جامعة ظفار خلال الفترة من 14 إلى 16 سبتمبر الجاري، بالشراكة مع وزارة الثروة الزراعية والسمكية وموارد المياه، بمشاركة نخبة من العلماء والباحثين والخبراء الذين يمثلون 17 دولة من مختلف أنحاء العالم. ويسعدنا بشكل خاص أن نرحب بمعالي الأستاذة الدكتورة رحمة بنت إبراهيم محروقية الموقرة راعية هذا الملتقى، مقدرين دعمها ورعايتها لمسيرة التعليم العالي والبحث العلمي في السلطنة. أيها الحضور الكرام، تولي جامعة ظفار البحث العلمي والابتكار مكانة محورية في رسالتها الأكاديمية ورؤيتها المستقبلية، إدراكا منها بأنهما ركيزتان أساسيتان في بناء مجتمع المعرفة وتأسيس تنافسية سلطنة عمان في ظل مستهدفات رؤية عمان 2040. وتسعى الجامعة إلى تهيئة بيئة أكاديمية محفزة لإنتاج المعرفة ونقلها وتوظيفها في معالجة القضايا التنموية وخدمة المجتمع. ولتحقيق ذلك توفر الجامعة الدعم المؤسسي والمالي واللوجستي للباحثين وأعضاء الهيئة الأكاديمية والطلبة عبر تمويل المشاريع البحثية وتبني المبادرات المبتكرة وتشجيع النشر في المجلات العلمية المرموقة، إلى جانب عقد المؤتمرات والندوات وتطوير الشراكات البحثية مع مؤسسات وطنية ودولية. إن هذا الاهتمام المنهجي بالبحث العلمي والابتكار يجسد التزام جامعة ظفار بترسيخ دورها كبيت خبرة وطني يسهم في رفض الاقتصاد المعرفي وصياغة حلول عملية للتحديات المعاصرة. ويأتي انعقاد هذا الملتقى في نسخته الثالثة ليجسد لنا ذلك الاهتمام بالبحث العلمي والابتكار من قبل جامعة ظفار. الحضور الكريم، إن اجتماعنا اليوم في رحاب جامعة ظفار ليس احتفاء بتراث عريق فحسب، بل هو التقاء بين أصالة التاريخ وعمق العلم الحديث، حيث يجتمع الباحثون والخبراء من مختلف القارات ليفتحوا صفحات جديدة في دراسة اللبان والنباتات الطبية. لقد تحول اللبان الذي كان يوما سلعة عابرة للقوافل إلى موضوع بحث علمي عالمي يختزن إمكانات علاجية ودوائية أثبتتها المختبرات الحديثة وتؤكدها الشراكات البحثية المتنامية. ولذلك فإن هذا الملتقى يأتي ليجسد رؤية علمية راسخة، رؤية تسعى إلى تحويل الإرث البيئي والثقافي لمحافظة ظفار إلى معرفة قابلة للتطبيق، وإلى ابتكارات دوائية وزراعية تضع السلطنة في مصاف المراكز العالمية في مجالات الطب الحيوي والاستدامة. أيها الحضور الكريم، تفخر جامعة ظفار بأنها لم تقف عند حدود البحث العلمي الأكاديمي فقط، بل خطت خطوات عملية في تطوير منتجات مبتكرة من اللبان، حيث تمكنت كوادرها البحثية من إنتاج 12 منتجاً متنوعاً على شكل أقراص وكبسولات ومراهم. كما تعمل الكوادر البحثية بالجامعة على تطوير المزيد من المنتجات الفريدة غير الموجودة في الأسواق العالمية، إسهاماً في علاج أمراض مرتبطة بالجهاز العصبي والمناعة. وقد أظهرت الأبحاث الأخيرة نتائج مبشرة للغاية تؤكد أن اللبان العماني مرشح بقوة ليكون مادة فعالة في علاج بعض هذه الأمراض. أعزائي الباحثين، إن هذا الملتقى ليس مجرد لقاء علمي، بل هو منصة للتكامل والتعاون وبوابة لتلاقح الأفكار والابتكارات بما يخدم أهداف البحث العلمي ويبرز الدور الحضاري للسلطنة كأرض اللبان ومركز للمعرفة والابتكار. ختاماً، أجدد شكري وتقديري لمعالي الأستاذة الدكتورة رحمة بنت إبراهيم المحروقية الموقرة على رعايتها الكريمة، ولوزارة الثروة الزراعية والسمكية وموارد المياه على شراكتها الفاعلة ودعمها الكبير لإقامة هذا الملتقى. والشكر موصول لغرفة تجارة وصناعة عمان فرع محافظة ظفار وشركة بيت البخور على رعايتها للملتقى، وكذلك الشركات الراعية الأخرى. ولا يفوتني أن أشكر كافة الباحثين والمشاركين والمنظمين على جهودهم القيمة، راجيا لكم ملتقى مثمرا وحوارا علميا غنيا ومقاما طيبا في ربوع محافظة ظفار. والسلام عليكم ورحمة الله وبركاته.
أيها الحضور الكرام، نشكر الأستاذ الدكتور عامر بن علي الرواس رئيس جامعة ظفار على كلمته، ويشرفنا الآن أن ندعو المهندس راشد بن سعيد الغافري مدير عام المديرية العامة للثروة الزراعية والسمكية وموارد المياه بمحافظة ظفار لإلقاء كلمته بالنيابة عن اللجنة الفنية المشتركة بين وزارة الثروة الزراعية والسمكية وموارد المياه وجامعة ظفار. فأهلا وسهلا به.
I'm honored to invite Engineer Rashid Saeed Al-Ghafiri, Director General of Agriculture, Fisheries and Water Resources, Dhofar Region, to deliver his address on behalf of the Technical Committee between the Ministry of Agriculture, Fisheries and Water Resources and Dhofar University.
بسم الله الرحمن الرحيم. الحمد لله رب العالمين، والصلاة والسلام على أشرف الأنبياء والمرسلين سيدنا محمد وعلى آله وصحبه أجمعين. معالي الدكتورة رحمة بنت إبراهيم المحروقية الموقرة، وزير التعليم العالي والبحث العلمي والابتكار، راعية هذا الملتقى، أصحاب المعالي المكرمون، أصحاب السعادة، حضورنا الكريم، يسعدني ويشرفني أن أرحب بكم في هذا الملتقى نيابة عن رئيس وأعضاء لجنة التعاون بين وزارة الثروة الزراعية والسمكية وموارد المياه وجامعة ظفار، والتي تضم في عضويتها مجموعة من شركات القطاع الخاص. هذا الملتقى الذي تنظمه جامعة ظفار بالشراكة مع الوزارة ومشاركة نخبة من الباحثين والخبراء من داخل سلطنة عمان وخارجها، له دلالة واضحة على الأهمية العلمية والاقتصادية والثقافية لهذا القطاع الحيوي. أيها الحضور الكريم، إن الشراكة بين الوزارة وجامعة ظفار من خلال لجنة التعاون المشترك تمثل نموذجاً رائداً للتكامل بين البحث العلمي والتطبيق العملي. وما يميز هذه اللجنة هو أنها لا تقتصر على مؤسسات التعليم العالي والبحث العلمي والقطاع الحكومي فحسب، بل تضم شركات ومؤسسات من القطاع الخاص، مما يوسع دائرة التعاون ويمنح الطلبة فرصاً للتدريب العملي الميداني، ويربط البحث العلمي بالجانب التطبيقي والإنتاجي، وبذلك تتكامل الجهود الأكاديمية والبحثية مع متطلبات سوق العمل في انسجام مع الأهداف التنمية الوطنية. وقد أثمرت هذه الشراكة الموسعة عدداً من الإنجازات المهمة، من أبرزها حصول جامعة ظفار على موافقة لتمويل مشروعين في مجال البحث والتطوير لمنتجات اللبان ضمن الدورة الرابعة للبرامج الاستراتيجية للوزارة بالتعاون مع وزارة التعليم العالي والبحث العلمي والابتكار. العمل على اعتماد مختبر فحص اللبان بجامعة ظفار ليكون مركزاً يقدم خدماته لمؤسسات القيمة المضافة لهذا القطاع. التعاون في مجالات البحث العلمي المرتبطة باللبان والنباتات الطبية والعطرية، وتنظيم المؤتمرات واللقاءات العلمية. دعم الكفاءات الوطنية، حيث يواصل عدداً من موظفي الوزارة دراساتهم العليا في جامعة ظفار مع التوجه لاستحداث تخصصات جديدة في مجال الموارد الطبيعية. تعزيز التعاون في مجالات استزراع الطحالب الدقيقة ومكافحة النباتات الغازية تكاملاً مع الجهود الوطنية في التنمية والاستدامة البيئية. تنظيم برامج تدريبية لطلبة الجامعة في مرافق الوزارة وشركات القطاع الخاص، إلى جانب مشاركة موظف الوزارة في المؤتمرات والورش العلمية التي تستضيفها الجامعة. حضورنا الكريم، نتيجة لما تحقق من إنجازات علمية في منتجات القيمة المضافة التي يدخل في مكوناتها اللبان والسوق الواعد لهذا القطاع، زاد الطلب المحلي والعالمي على اللبان العماني، فعملت الوزارة بالتعاون مع الجهات المختصة على التوسع في زراعته عبر إنشاء مزارع متخصصة، حيث تمت الموافقة على 17 فرصة استثمارية، ومن المتوقع زراعة أكثر من حوالي 92 ألف شجرة لبان جديدة على مساحة حوالي 1.5 مليون متراً مربعاً خلال الفترة القادمة. معالي الدكتورة راعية المناسبة، حضورنا الكريم، في الختام لا يسعني إلا أن أتقدم بالشكر والتقدير إلى معالي الدكتورة راعية المناسبة وإلى جامعة ظفار على استضافتها لهذا الملتقى، وإلى كل من حضر وشارك من داخل سلطنة عمان وخارجها، متمنيا التوفيق والفائدة للجميع، ونسأل المولى عز وجل أن يعيننا على تحقيق الخطى الملهمة لهذا الوطن العزيز، وأن يديم الأمن والسلام ويعم كافة الأقطار، وأن يجمعنا دوما للتعاون بما يخدم البشرية ورقيها واستدامة مواردها. والسلام عليكم ورحمة الله وبركاته.
نشكر المهندس راشد بن سعيد الغافري، مدير عام المديرية العامة للثروة الزراعية والسمكية وموارد المياه بمحافظة ظفار على كلمته. والآن يسعدنا أن ندعو الأستاذ الدكتور لؤي رشان، مدير مركز الأبحاث بجامعة ظفار، لإلقاء كلمته بعنوان استكشاف واستخدام اللبان العماني وبعض النباتات المختارة من أجل التنمية المستدامة في عمان، فليتفضل مشكوراً.
Now, I have the pleasure of calling upon Professor Lؤي Rashan, Director of Research Center, to deliver his keynote speech. Professor Lؤي will present his keynote speech about exploration and utilization of Omani frankincense and some selected plants for sustainable development in Oman.
بسم الله الرحمن الرحيم، والصلاة والسلام على سيد المرسلين محمد صلى الله عليه وسلم تسليما كثيرا. Excellency, Minister of Higher Education, Scientific Research and Innovation, Professor Dr. Rahma bint Ibrahim al-Mahruqiyya, Excellencies, guests, staff members, students, it gives me immense pleasure to be with you today, celebrating this very important event. This event is about the third international meeting on frankincense and medicinal plants. I hope that this meeting, which is organized by the Dhafar University and in collaboration with the Ministry of Agriculture, Fisheries and Water Resources, will reach into the plans already, will reach into the target plans. Also, I would like to take this opportunity to thank delegates from more than 17 countries. We have delegates from all over the world, and in this meeting, we are going to deliver and give more than 65 talks. All these talks will be focused on frankincense and other medicinal plants. Finally, again, I would like to thank Her Excellency for visiting and for opening this ceremony. And also, I would like to thank very much Ministry of Agriculture, Fisheries and Scientific Research for their kind support. And I think our long-standing relationship will grow and grow, and in order to achieve the long collaboration and in order to reach into the target. Now, allow me please to present my talk. And as you can see, my talk, and the title of my talk is different from the talk which was presented at the second and the first conferences, the first and the second meeting. And as you would see, in this meeting, I am going to, at the end of my talk, to focus on very important achievement, which the Far University and in collaboration with more than 20 international universities and centers achieved. Well, first of all, allow me to say something, As you know, Sultanate of Oman is very rich, especially the far in biodiversity of both flora and fauna. Research Center was established in the year 2016, and the establishment of this center is articulated on three main angles. The first angle is to investigate and study the chemical, biological, pharmacological, and toxicological of many endemic plants which are present in the Far. All in Oman, there are more than 1,200 species of plants, but 850 of these plants are growing in the Far. 65 of them, they are endemic, and the most important tree, which is well known all over the world, is frankincense tree. Second, to investigate the possibility of potential activities of these plants as potential therapeutics. And finally, our goal was and is still to develop and formulate scientific evidence-based products that can be described at this stage as food supplement and cosmetic. But in stage 2, as I will explain in a few minutes, we are planning to move ahead and to go beyond this capability, and we are planning to develop and formulate other products like herbal preparations, herbal drugs, orphan drugs, and also you may be, I will update you about some other interesting issue which we achieved in the last 3 years. This is the tree of frankincense, as you can see, and this is the way how frankincense is harvested. And these are the grades, resin grades, which I call them grades and not types. All of them, they are produced by one tree, although the color of the gum resin varied depending on where the tree is growing. However, all these gum resins, they have the same chemical composition. All of them, except they may vary, but this variation is not significant in the amount of the chemical components which are present in the chemical resin, in the resin itself, sorry. Now, I can say that after the establishment of the research center, we prepare a study, and this study can be divided into 2 phases. Phase 1 is from 2016 to 2022. In this phase, we studied the phytochemical, biological, pharmacological, and also we define and study the safety profile of the gum resin as well as some of the extract plus the essential oil. And then in the third stage, we started developing and formulating some products. These products, as I said, can be classified as food supplement. They are either capsule or tablet dosage form. Also, we have liquid dosage form in addition to the cosmetic. Again, cosmetics can be graded into ointment, creams, lotions, and other dosage forms. Now, we are preparing the documents according to the guidelines of the Ministry of Health in order to submit and approve them as scientific evidence-based herbal products. These are some of the products which we developed. Their number is 10. However, we developed 2 additional products. I'm sorry, I don't have the artwork of this product to show, but these 2 products, the last 2 products which we developed, they are very important, and they can be used to support the nervous system. Now, phase 2, from 2022 to 2026. And I can say, and most of you know, that most of the diseases in a human being can be attributed to a phenomenon, can be described as inflammation. Neurological diseases and neurodegenerative diseases, there are more than 100 or even more of these diseases. All of them, they are caused by inflammation. The first step, cancer. There are more than 150 types of cancer, and all of them, they start by the process of inflammation. Diabetes type 2, again. Therefore, I can say at the present time that we have very important data regarding neurodegenerative diseases, and we have 2 publications so far published last year and this year. And the first publication was about brain trauma, and the second project which we published about brain protection. We have other manuscripts in the preparation about myofibrils, plus we have another product about autism. Also, we have significant data about cancer. We completed all in vitro and in vivo, and we are moving now to study the molecular mode of action of how these extracts and gum resin produce activity. Same can be applied to diabetes. We have 1 manuscript published, and we are planning to study the molecular mechanism or the molecular mode of action of this product. At the present time, we are focusing on plant extracts, واللي في الوقت الحالي تسمى مستنبطات. We have more than 40 extracts obtained from frankincense. However, we are focusing on 2 at the present time. 2 extracts. One of them, I will name it now as agro or green extract, and the other one, I can name it as the standardized extract, which is obtained by an organic solvent. Also, we managed to isolate some pure compounds, not from boswellia, but from other medicinal plants. And these products which we identified, we are evaluating them, and in the second step, we are going to prepare derivatives, chemical derivatives, and finally, I can say that we synthesized in our labs, research center, more than 12 new derivatives from a molecule that isolated from a third, a plant growing in the far. Agro. What is agro? What is green extract? Agro or green extract, this approach is used recently since the year 2022, as I learned from my colleague, Professor Soleimani from Loran University, and he will tell you more, in the first session after the closing ceremony. And in this project with Loran University, we obtained very good results in which we obtained 4 or 5 types of granules from the gum resin. They are classified according to their microns. And we know now and identify which granule contain the highest amount of benzoic acids, the main functional compounds which are responsible for inhibiting all anti-inflammatory or inflammatory components. Now, many companies which are mainly interested in food supplement, they are looking for agro products. Why agro products? These agro products also, they can be named as eco-friendly. No solvents will be used. Therefore, there is no toxic substances in this extract, in addition to no waste, low cost, produce highly pure extracts, and valorize agro-industrial waste into valuable products. Second type of extract, which we are, planning to reach into our last step. We are trying since 2 years ago to standardize this extract. Now, standardization is very complicated procedure. And in order to do it, we have to do it at GMP facility in order to be identified by pharma and other companies. Second, we have to study the safety assessment. And studying safety assessment should be done at a GLP approved company. On the other hand, there are very limited studies regarding the pharmacokinetics, PK, of frankincense. Only very limited. And this is a very important issue, and we are going to start these studies soon. In addition to no studies available regarding bioequivalence study in Luban, neither in Omani Luban nor in other Lubans, which are grown in other countries. Again, we are preparing to study this phenomenon on 22 patients. Now, we have to do also complete scientific validation of frankincense efficacy in order to produce another category of dietary supplement, which is called functional or novel food. And there are many novel foods or functional foods now in US and other European market. And we will start this project very soon again. And also we need to have testimonies and endorsement from practitioners and key investigators. Now, again, I would like to emphasize on having the following in order to have strategic pillars in order to get potential activity of standardized extract. We have to follow all these categories, which I nominated some of them a few minutes ago. Now, we have a request to the Ministry of Agriculture, Fisheries and Water Resources, Environmental Authority, and other government bodies to ensure sustainability of the endemic plants, such as frankincense tree and other important endemic plants, by legalization certain laws, by formulating laws to protect these plants, especially those plants which we are focusing now on studying these plants. And finally, this is the last slide before one. So far, we have 2 granted patents, 1 international, 1 local. We filed US and European patent. We have another patent in preparation. We published more than 70 manuscripts, and we have more than 10 ready to submit. And also, we have done some limited case study, I cannot say clinical studies, and in collaboration with our colleagues in Switzerland. He is going to give a talk, and he is a medical doctor, and he also conducted many studies, not only on cancers, but also on cryptogenic pneumonia, and also on some other neurodegenerative diseases. Now, I have good news to tell. The first news which I would like to tell that Dhafar University Research Center is the first center so far worldwide to identify new compounds isolated from Omani frankincense, Boswellia sacra. 2 of these compounds, they have a structure which is similar to steroid compound. And let me say it in Arabic, please. هذه الظاهرة ظاهرة فعالية اللبان، لو رجعنا إلى كل الأبحاث المنشورة من 1950 لغاية اليوم التي تمت على اللبان الهندي واللبان العماني واللبان الصومالي والأفريقي وغير ذلك، تشير إلى وجود مادة غريبة في مادة اللبان تعمل عمل الكورتيزون، لكن تركيبها غير معروف، شكل وزنها الجزيئي غير معروف. احنا في هذا المركز وفي هذه الجامعة وبالتعاون مع بعض الجامعات we managed to identify 5 new compounds, and other informations will be disclosed, soon after we file a patent about these compounds. Second, we managed to synthesize a new compound, which we named it as wood-fordiomycin. This compound isolated by a scientist from South Korea, and he is one of our keynote speakers. And synthesis was achieved by ours. They isolated this compound. They investigate the potential activity of this compound against many fungi, and they compared it with the standard drugs. And because this product is very important, we did synthesis to this compound, and we are planning also to synthesize new derivatives from this compound. Again, and also we have more than 12 new compounds isolated from another plant growing in Dhafar, and we did almost anti-cancer, antibacterial, antiviral. Also, we did some tests on its effect on neurodegenerative diseases, and we are going to move further in order to evaluate these compounds. In addition to, we have other projects in progress. Thank you very much indeed for your careful listening, and allow me now to introduce our first keynote speakers, Dr. Ian Cook from Griffith University. Thanks to Dr. Ian for coming and attending our conference. Dr. Ian Cook obtained his PhD and bachelor degree from Griffith University. He is working now as senior lecturer in biochemistry at School of Environment and Science in Australia, Griffith University. He is member in the editorial board of more than 7 journals. He published more than 300 articles. Also, he published some books. Finally, he graduated many master and PhD students. Welcome to Ian Smith. Please, the floor is yours.
I'm not sure how to switch on my talk. It's one of the technical... It's not up there, though. Okay, thank you, Dr. Rashan, for the wonderful introduction. I'd also like to thank you and the organising committee, as well as the sponsors of this meeting, for inviting me here to give this talk. This is my third time in Salalah at these meetings, and it's good to come back to see how well things are progressing. In the previous two meetings when I spoke, I always spoke on Boswellia sacra and the work that we were doing on this plant. Today, though, I'm going to be speaking on a different plant that's also native to this region, and I think it deserves a lot more attention than it's actually getting. In particular, I'm going to be talking about Woodfordia unifloria and its healing properties on wounds and on gangrenous ulcers. So this plant is a really interesting plant. It's used in traditional medicine for a variety of different purposes. It's used in treating rheumatism and inflammation. It's used in gastrointestinal problems such as diarrhea and dysentery. But most importantly for the talk that I'm going to be giving today is that it's very useful for skin diseases and aid in wound healing, particularly of gangrene ulcers. So you probably know with gangrene, it's largely caused by the bacterium Clostridium perfringens when there's restriction in blood flow in a specific area. And it can cause some really major problems. It can result in tissue destruction, which can result in amputation, or if not treated, can even result in death. Now, I'm going to be showing a slide in a second that actually shows a gangrenous ulcer. So if you think that may upset you, you might be advised to turn away for a minute or two, and I'll tell you when you can turn back again, because it is a reasonably graphic picture of a gangrene ulcer. Or not. Oh, there we go. This is a photo that Professor Rashan sent to me a couple of years ago that got me interested in this plant. Basically what we see is on the left-hand photo here, and the pointer isn't working, but that's okay. On the left-hand photo, we actually see a gangrenous ulcer, and it was treated with the preparations from Woodfordia unifloria, and as you can see, there's healing occurring there. So it does actually work in a clinical setting. So we set out to actually determine what is going on there, and the first thing that we needed to look at was the ability of this plant to inhibit the growth of Clostridium perfringens, because this is the major cause of these gangrenous ulcers. To do this, we used two different types of assays to start out with. We used, on the left you'll see agar diffusion assays. This is a preliminary test that we use to look at the susceptibility of the bacteria to the preparations. And we also use liquid dilution assays that's shown on the right to actually quantify the strength of the response. And this uses a molecule that's shown up there, which is iodo-nitrotetrazolium salt. It's easy to assays to do because it changes colour in response to actively growing bacteria. So you can see where there's bacteria present, that we have the red colour change, and where there's no bacterial growth, it remains uncoloured. So we first tested the growth of Clostridium in the agar diffusion assays, and again I don't have the pointer, but the bars on the right, the light coloured orangey sort of bars on the right, show the positive controls that we used, various different antibiotics, and these were high doses of antibiotic that we used so that we were sure that we would get a response. The bluey coloured bars and the green and the grey bars are the different extracts that Professor Rashan provided us with. And as you can see, particularly the ethanol and methanol, the blue bars on the left, they gave us very good zones of inhibition. They may look small compared to the the positive controls, but again, the positive controls, we use massive doses to be assured that we would get a response. So from this, it shows that ethanol and methanol extracts in particular were very good at blocking the growth of Clostridium perfringens. Now from this table, I'd like you just to narrow in on what I've got circled in red. These are the results of the liquid dilution assays, the quantitative results that we performed. And hopefully you'll be able to read that there are some very good MIC values there. We had MICs down to 250 micrograms per mil, which is actually quite good, quite potent for Clostridium perfringens, which shows that yes, these extracts are actually quite good at inhibiting the growth of Clostridium perfringens, the bacterium that largely causes gangrene and gangrenous ulcers. However, when we're talking about healing these ulcers, it's more than just stopping the growth of the causative bacteria. We need to be aware that there's, in healing the wounds, there's a number of different phases of wound healing. The left-hand panel, I'll have to refer to them by the panels because I don't think the pointer's working, but the left-hand panel labelled homeostasis shows the initial events in wound healing. So when we've got the Clostridium perfringens causing the ulcer, we can get other bacteria getting into that as well, causing infections, and that will actually hamper wound healing. So we need to be able to control bacterial infections. Moving on to the second panel, inflammation is also important in wound healing. It is actually a necessary process, but if it gets out of control or if it's too severe or chronic, it can delay wound healing as well. Then there's proliferation. What we tend to think of when we think of wound healing is the cells being replaced and the wound closing up. And finally, remodelling. So if we're looking at wound healing, we need to consider all of these phases. So initially, after we found that the plant extracts worked well against Clostridium perfringens, we decided to look at other bacteria that can cause infections in these wounds, and they would hamper the healing process. There's a lot of different bacteria that can actually be involved in this, but the main ones are your normal skin microflora, Staphylococcus and Streptococcus species in particular. We have tested others as well, but I'll focus on these ones for today. Okay, now what we have here is in the top left panel is the activity against Staphylococcus aureus. The top right panel is a methicillin-resistant strain of the Staph aureus. And in the bottom left, Staph epidermidis and Streptococcus pyogenes. And hopefully what you can see in all of these cases is that the extracts were effective, particularly the ethanol and the methanol extract. They were effective at blocking the growth of all of these bacteria. If the pointer worked, I would point out to you the top right panel in particular I found particularly interesting, because this is a very resistant strain of bacteria, and it worked just as effectively against this strain of bacteria as it did against the normal Staph aureus. So it's bypassing some of the antibiotic resistance mechanisms, which I think is very interesting for these extracts. And again, when we quantified these results, we again, for all of the bacteria, the methanol and ethanol extracts in particular, gave us very good MIC values, around 250 micrograms per mil, which is very good druggable targets. So all of these were strong against these bacterial species that are involved in the wounds as well, meaning that we could block the first phase or enhance the first phase of wound healing. The second phase is inflammation. And inflammation, I will say, it is a necessary event in wound healing. You do need inflammation to occur. It brings the cells, many of the cells involved in wound healing to the site. But if the inflammation is too strong or it goes on too long, it can actually hamper the wound healing. So the ability to regulate inflammation is also important in wound healing as well. So we decided to test the extracts against various events in inflammation, and I will say at the outset, inflammation is a really, really, really complex process, way more complex than this figure makes it look. This is just a bit of a snapshot. So we decided to actually focus on one process of inflammation, or one part of inflammation, which is the production of eicosanoids, particularly prostaglandins, that are involved in inflammation. So when you have an inflammatory response, say infection in the wound, arachidonic acid will be released from cell phospholipids. That arachidonic acid can then be acted on by either cyclooxygenase enzymes or lipooxygenase enzymes, resulting in the production of inflammatory mediators, prostaglandins and leukotrienes in particular. We focused in on the prostaglandin pathways, which are mediated by the enzyme cyclooxygenase, or COX. We specifically focus on COX-2 because that is the form that's induced during inflammation. And we also looked at, excuse me, prostaglandin E2, which is one of the main mediators of inflammation. Now, to look at these, we used commercial ELISA assays, which are quite simple. They are 96-well plates that have antibodies to the molecule that you're looking at attached to the base. You put your sample in, add another antibody with a colorimetric molecule or an enzyme on it, and you can actually measure and quantify the change based on the colour change. Now, using these assays, again, the right-hand bar labelled NC is our untreated control. The TU is turmeric, which is our positive control. All of the others are the extracts. And hopefully what you'll see, particularly the ethanol and methanol extracts, they were very strong inhibitors of cyclooxygenase 2, although there was good inhibition in the water extracts as well. We also looked at inhibition of prostaglandin E2, and again, we mirrored the same results. Again, there was very good inhibition of the prostaglandin E2 by the methanol, ethanol, and also the water extracts, meaning that we could actually block many of the main mediators of inflammation. Okay, the next phase of wound healing, though, is the tissue growth, the proliferation of the cells. So we also looked at the ability of these extracts to affect fibroblast growth, because that is where the cells are actually replaced during inflammation. Now again, using these assays, again on the right-hand bar, that's our untreated control, and the line across the horizontal line shows where the 100% response is, the response of the negative control. And hopefully what you'll see, particularly again with the ethanol and methanol, but also with the water extracts, there was very strong stimulation of fibroblast growth, meaning that we could actually stimulate healing of this wound by replacing the tissue at a much more rapid rate than without. The fourth stage of wound healing is it's a remodelling. We haven't actually looked at that one yet. We need to develop an extracellular matrix model to look at it, but that is something that we aim to look at sometime in the future as well. However, what we have shown is that these extracts are essentially hitting 3 of the 4 of the phases of wound healing and enhancing them, meaning that they would be good for all phases of wound healing that we've tested. Now, to be effective as a drug, these also need to be relatively non-toxic as well. We have done other toxicity assays. I don't have time to go into all of them, so I'll just draw your attention back to the fibroblast assays that showed that not only were these extracts not toxic, they actually enhanced cell growth. We have done other toxicity assays too, but I just don't have time to go into those today, unfortunately. So to summarise what we found, the Woodfordia unifloria extracts were strong inhibitors of Clostridium perfringens growth. They also inhibited the growth of other bacteria that are involved in wound infection as well. So they affect the first phase of wound healing. They enhance the ability for you to heal the wound by attacking those phases. They also inhibit cyclooxygenase 2 and prostaglandin E2 production, meaning that they can be used to modulate the inflammatory phase, and they also stimulate fibroblast growth, meaning that they would actually positively affect 3 of the 4 phases of wound healing. I therefore think that this plant is a really interesting plant that deserves a lot more attention than it's currently got, and I assume our Korean delegate's going to be talking more about this one in other senses from what Professor Lue mentioned a second ago about his work on this plant or the molecules as well. I'd like to finish up by acknowledging Professor Louay Rashan, who got me interested in this plant to start off with, and is collaborating on this, and on Dhofar University for having me here and also being involved in the project. Thank you.
نشكر الدكتور إيان إدوين كوك على كلمته، وندعو الآن معالي الأستاذة الدكتورة رحمة بنت إبراهيم المحروقية الموقرة وزيرة التعليم العالي والبحث العلمي والابتكار، والأستاذ الدكتور عامر بن علي الرواس رئيس جامعة ظفار أن يتقدما إلى المنصة لتقديم هدايا الشكر والتقدير للرعاة والعارضين على التزامهم ومساهمتهم.
We will now request Her Excellency Professor Dr. Rahma bint Ibrahim al-Mahrouqiya, Minister of Higher Education, Research and Innovation, and our Vice Chancellor, Professor Amr bin Ali al-Rawas, to come up to the stage to present a token of thanks to sponsors and exhibitors for their commitment and contributions.
أولاً الرعاة الرئيسيون وزارة الثروة الزراعية والسمكية وموارد المياه.
Our main sponsors, Ministry of Agriculture, Fisheries and Water Resources.
الرعاة البلاتينيون بيت البخور العالمي للاستثمار الصين.
Platinum sponsors, Global Incense House, China.
غرفة تجارة وصناعة عمان فرع ظفار.
Oman Chamber of Commerce and Industry, Dhofar branch.
الرعاة الفضيون شركة مكة للتجارة.
Silver sponsors, Mecca Trading.
لبان ظفار.
Silver sponsor, Luban Dhofar.
ثانياً العارضون. حملة الصيدلية الخضراء من وزارة الصحة.
And now the exhibitors, Green Pharmacy Campaign by Ministry of Health.
مركز أبحاث جامعة ظفار.
Dhofar University Research Center.
بيت البخور العالمي للاستثمار الصين.
Global Incense House China.
لبان ظفار. جزيرة اللبان.
Luban Island.
مكة للتجارة.
Mecca Trading.
العماد لإنتاج الزيوت العشبية.
العماد for herbal oil production.
تبوك للأدوية.
Tabuk Pharmaceutical.
أيها الحضور الكرام، يتقدم الأستاذ الدكتور عامر بن علي الرواس رئيس جامعة ظفار بهدية تذكارية إلى معالي الأستاذة الدكتورة رحمة بنت إبراهيم المحروقية الموقرة وزيرة التعليم العالي والبحث العلمي والابتكار لتفضلها برعاية هذا الحدث.
And now, Professor Dr. Amir bin Ali Al-Rawas will present a gift of gratitude, Excellency Professor Dr. Rahma bint Ibrahim Al-Mahrouqiyya, Minister of Higher Education, Research and Innovation.
ندعو الآن معالي الأستاذة الدكتورة رحمة بنت إبراهيم المحروقية الموقرة وزيرة التعليم العالي والبحث العلمي والابتكار للتفضل بقص شريط المعرض المصاحب لهذا الحدث، والذي يليه استراحة الغداء والعروض المصاحبة للحدث في القاعتين 1 و2، والتي تعد فرصة رائعة للتواصل، تبادل الأفكار والمشاركة في أحدث الأبحاث المتعلقة باللبان والنباتات الطبية.
We will now request Her Excellency Professor Dr. Rahma bint Ibrahim Al-Mahrouqiyya, Minister of Higher Education, Research and Innovation, to cut the ribbon for the exhibition, accompanied by lunch break and presentations in respective track in Lecture Hall 1 and 2. This is a wonderful opportunity to network, exchange ideas, and engage with the latest research in frankincense and medicinal plants. Wassalamu alaikum wa rahmatullahi wa barakatuh.